Here at CoolLED today with Dr. Martin Bootman. Dr. Bootman, please welcome.
Thank you.
Dr. Bootman is a Director of Research at the School of Life, Health and Chemical Sciences at the Open University. He's a long-standing interest in the dynamics of calcium signals and the mechanisms by which the body interprets those signals. He's authored or co-authored over 130 publications in the past 28 years, and most of those have involved calcium signaling. Dr. Bootman, can you tell us a little bit more about calcium ions and what they actually do? Why are they important?
Sure. Yeah, and absolutely. So it's been a fascination of mine ever since I was an undergraduate student, actually, how cells regulate calcium and how they use it as a messenger. As an undergraduate, I did a project working in a lab where they were interested in lens opacities, things that led to cataract, for example, and it was calcium. It was actually precipitates of in all aspects of life. So right at the beginning of life, when a sperm meets an egg and there's a fertilization event to create an individual, one of the first things that happens is a calcium signal. And so the sperm comes along and it binds to an egg, and it actually causes a calcium change inside the egg, and that calcium change is necessary for several things. It initiates the developmental program because the egg is arrested, suspended animation, if you like, waiting for that sperm to come along and reactivate the cell cycle, as it's called, so it can divide and turn into an individual. And the sperm coming along and causing this calcium signal as part of that activation. It also lifts what's called a fertilization membrane. This stops other sperm coming in because, of course, the egg only wants to fuse with one sperm, so you have the combination of genetic material that you need for an individual, but no more. And so the interaction with the sperm and the egg causes this calcium signal, which in mammals, such as humans, causes a series of calcium oscillations, and those oscillations are absolutely necessary to drive development. So right at the beginning of life you have calcium. And then unfortunately right at the end of life, causing cell death and causing fatalities and pathological conditions, you also have changes in calcium. But these of course would be aberrant changes in calcium. So in conditions such as cancer or Alzheimer's disease, you have changes in calcium that you don't want. And of course, all the way through life as we're well, muscle contraction, memory formation, cell proliferation, that's all driven, at least in part, by calcium. There are very few processes inside cells in which calcium doesn't play a role. And so that's really the fascination. No two cell types use calcium the same way. They use calcium, but not in the same way. So the spatial dimension of the calcium signal, the kinetics of the calcium signal, how it spreads through the cells, the particular channels that transport calcium into aspects of the cell, they'll be different, but calcium will be in there. And that's what keeps the many thousands of people who work on calcium around the world interested, is how do different cell types use the same messenger to control so many different things, and how is the signal manipulated in such different ways. And whilst we understand quite a lot about how cells use calcium, we still have an awful long way to go, particularly in those conditions such as cancer and Alzheimer's and various diseases like that I mentioned before. We still don't quite know what's gone wrong to cause those conditions. What's the subtle change where you've gone from a physiological condition in a neuron to one that's a pathological condition where that neuron is not forming memories properly anymore? What's happened? And what it seems to be is just a modest elevation of calcium at the wrong time can wipe all your memories, and that seems to be a partial explanation for why there's poor memory formation, for example, in Alzheimer's. Just a subtle change in the calcium level inside cells.
And calcium is the messenger?
Yes, so...
how does that affect the detection, and how do the different operations of the cells affect the detection and signaling of calcium? So calcium can get into cells or be released from stores inside cells in lots of different ways. And cells have kind of a toolkit. That's how we think about it. All sorts of different elements.
And just as you would have a toolkit in your garage, which might enable you to do all sorts of jobs around your house, in terms of detection, in terms of trying to understand the calcium signals, we need to know something about the physiology of those cells. So if they have fast calcium signals, which neurons might do, or if they have slow calcium signals like epithelial cells might do, we would need a different kind of detection system. We would need to consider the calcium changes occurring in a different region of the cell, perhaps, and having a different readout, so a different physiological role. So the many different types of tissue that make up an individual, they do use calcium, as I said earlier, but they use it in very different ways. So the detection the method of detection that trying to understand what the calcium signal does, its role as a messenger, that very much depends on the tissue type.
And can you talk about some of the equipment that's used to detect the calcium?
Right. So there's a whole range, actually. So when I started my studies as a PhD student many years ago, we used to do a lot of radioactive calcium work. We used to try to follow calcium moving through cells using calcium-45, which is not particularly dynamic, of course. It was slow and cumbersome, not to mention expensive, and somewhat. I mean, working with radioactivity, of course, is not ideal. A lot of what's done these days involves fluorescence. So when I started my PhD studies many years ago, there was a company called Molecular Probes who had a few fluorescent compounds, a few markers for particular cellular organelles, for example, and a few calcium indicators. But it was really a pamphlet in those days, and their handbook of fluorescent probes grew and grew over the years, and now, of course, it's so large that it's rarely printed. It's just looked at online. And that's a testament to the fact that fluorescence has grown and grown and grown as a way of investigating cells in a non-invasive kind of way, in a way where the cells are still vital, they're still alive, and you can watch them do what they would normally do. You can watch them functioning in a physiological sense. Or, if you've taken cells, like cancer cells, for example, you can study them in their genuine pathological state, if you like, as well. So I think whilst there are lots of other techniques that are used, for example, there's electrophysiology, calcium-45 is still used, electron microscopy is still used. I think the large growth area has been fluorescence, and in particular the utility of things such as fluorescent proteins, for example, for which of course the Nobel Prize was awarded just a few years ago. And these enable you to tag specific locations inside cells, specific proteins. You can look very dynamically at things that are happening inside cells. So most of what we do is based on microscopy, either wide field microscopy, where you look down and you see the whole of a cell, or confocal microscopy, where you can optically section through cells, which is very useful if you want very high resolution images of cell structures or processes that are going on. But there are other modalities with fluorescence as well. There are other things such as TIRF and FRAP and various things that could give you either a view of what's going on in very, very small subcellular domains, or enable you to look at the mobility of proteins. And with the advent also of super resolution technologies as well, which get down below what's called the diffraction limit, we can really start to get a hugely improved view of the ultrastructure of cells. And again, some of these techniques are, not all of them, but some of these techniques are real-time techniques. So you can actually look at just incredible detail of signaling or structures inside the cells using fluorescence in living cells in real time.
Yeah. And there's lots of different markers that are used in fluorescence signaling. Can you talk about some of the more popular ones that
Absolutely.
you've used?
Yes. So it's an essential thing that's happened over the past decades has been the ability to multiplex different indicators. The reason is because we understand more and more that about the interactions. So if you take calcium, for example, if there's a calcium signal, then that will have several cellular readouts. And in the past, we might have done an experiment where we imposed a calcium signal on side cells, or we used some natural mechanism to cause a calcium signal, and then we'd have done an assay afterwards to see what effect that calcium signal had. Well, given the advent of fluorescent proteins and the technologies to actually image those fluorescent proteins in living cells, we can now do these experiments simultaneously. So we can actually impose a calcium signal, and we can use another fluorescent marker to look at the readout. But very often, not just one readout. We can actually look at simultaneously at 2, 3, 4 readouts. This enables us to put a sequence of events together. So we're not doing one experiment in isolation from another and then kind of putting them back together again. What we can do now is we can multiplex these experiments using different fluorescent markers, which are specific for particular readouts. And I think that's an essential development that's happened over recent years. Also, the combination with different modalities of imaging as well, so you can combine wide field imaging with other modalities, so that you can actually look at the whole cell, see what's happening, for example, and then look in a specific region. You can do FRAP, or you can do FRET, or you can do TURF, and you can actually look at these things sequentially. We have the ability now to really follow different cellular processes in real time, with good resolution, spatially, temporally, where we can actually really start to put cause and effect together, if you like.
Have there been any specific developments that have enabled multiplexing that you've used that have helped you?
I think a few things. I think sometimes the technology changes in kind of almost quantum leaps, and I think the development of the fluorescent proteins, since the cloning of GFP, some years ago.
Which was the subject of Professor Tian, the
Yes, that's right. Yeah, yes, that's right. Yes, Roger Chen sadly missed, actually, who I was lucky enough to work with for a very short period of time and publish with. Really remarkable guy, actually. And of course, the founder of the calcium indicator dyes that we use now.
Yeah.
We owe him indeed a great deal. So the development of different coloured fluorescent proteins, simultaneous with the development of very sensitive detector systems, which enable those fluorescent proteins to actually be visualised inside cells, seems to me have gone pretty much hand in hand over the last couple of decades. So we have, as well as the development of non-protein based, fluorophores as well. So there's been, that increase that I mentioned before. For example, companies, the former molecular probes, and all the fluorescent reagents they have has gone hand in hand with developments in technology as well to enable us to illuminate those fluorophores. Yeah.
Is there an ideal detection system?
So I think most labs that use fluorescence would have a range of systems, and we certainly do. So some of the things that we study are over in seconds. Some of the things that we study take many, many minutes. So we need a different temporal modality, if you like. We also know that some of the processes that we look at, it's fine to get an average cellular view on what's happening. But for other aspects of the biology that we're interested in, we really need to know what's going on in very discrete areas of a cell. And so we use a combination, for example, of wide field imaging, confocal imaging, and, for example, TURF, if we need to get very close to the membrane. It really does depend on what particular aspect of biology or signaling you're trying to study. But most labs would actually have a range of technologies because we simply need to actually put the story together using a combination. There isn't really one technology that is all singing or dancing for everything that we need to do. You might start at a basic end with wide field imaging and then find yourself asking more and more precisely focused questions that then require you to go off to other systems.
As you go deeper and deeper into the cell.
I mean, it might be that those different imaging modalities are available on the same system, but generally not. But you still might need to switch between the different modalities to actually answer different questions.
And is there a particular fluorophore that enables you to do this in one swoop, or are you using different fluorescent proteins?
Yeah, so we use a whole battery of different things. I mean, we have some stock fluorophores that we use. With regard to calcium, we tend to use Fura-2 because it's just so widely used, and we've been using it now for a couple of decades, and it's just rock solid, in terms of its readout. It's so well understood, so well used. We also, but then Fura-2 is fine when we're doing wide field imaging. For confocal calcium imaging, we need to go to visible wavelength indicators, and we've used Fluor-4 and Fluor-8, and the Cal 520, Cal 590 we've recently moved on to because we find them to be more photostable. We're forever looking around to see the new indicators that come through. We very often try to get hold of a batch, give them a test, see if they have a better dynamic range. That's the change in brightness from calcium free to calcium bound, for example, whether they have more photostability or whether they're just better in our system. We're slow to adopt new ones unless there's a real reason to do it, which is why, for example, we've stuck with Fura for so many years. It works, it's rock solid, it's used universally, so it's hard to see us changing from that. But yes, we are very keen to know what's coming through. And there are new fluorophores, new proteinaceous fluorophores coming through all the time. And it's important to keep your eye on that because they might be a better way of tagging proteins or structures inside cells you're interested in. They might be better for particular applications. So you really need to keep an eye on those.
We talked a little bit about the methods and going deeper into the cells, talked a little bit about the dyes. What are the key, for you, the key elements of the setup that you're looking for in terms of possibly stability, reliability? You've talked about rock solid Fura-2 as a dye. Is there...
So I think the straightforward answer to that is that it's not often me that gets to do the experiments. So it's my PhD student who gets to do the experiments most often. And what I need to know, so that we can have surety, and confidence in the results, is I need to know that they're working with very robust systems, and that's very important.
Robust as in repeatability, is that the most important?
I would say repeatability, but also not just in a mechanical sense, but actually having that intuitive confidence in the data. We are our own worst critics. We have to be, and I always say to my students that if we can't convince ourselves of a result, we're never going to convince anyone else. And the only way we can convince ourselves of a result is that we perform those experiments multiple times under the best control conditions we can. And what we need is the technology that we use to capture cellular responses to be absolutely faithful in the way it does that. The cells are variable enough. There's quite a lot of variability in biology, and sometimes if we have a field of cells that we're imaging and we put on a particular hormone or an agonist that we know will generate a calcium signal, when we say we know, that'll happen in 95% of the cells. The 5% of the cells don't respond, and we don't know why, or we might not know why now. So there is quite a lot of variability, and even within those cells that do respond, the amplitudes will be very different, the temporal patterns will be very different. And these are things that we still study, they've been known for many years, but we still don't have a full answer to. So there's a lot of biological variability in the experiments we do. What we don't want is system variability impinging on that. We really need the systems that we use to be rock solid, because then we can understand that the variability that we're investigating comes from the system and not from the technology. And that's quite important to us. And also when I need to know that when my students bring me data, I don't have to quiz them about, was the scope behaving well today? Was this a good day of experiment? I have the assumption, because the technology we have is very mature and very steady, that with the same settings, with the same technology, the system would always give the same readout. So the variability comes from the cellular system, not from the imaging system.
And in your working career, you've seen the development and evolution of LEDs, which are CoolLED speciality. How are LEDs helping in achieving this stability that you're looking for?
So we've over the years we've used of various light sources. We've used arc lamps and we've used gas lasers, and we've seen a transition from gas lasers to solid state lasers, which is has been extremely helpful because they're just turnkey solutions. They're almost instant on. And so where solid state lasers have been helpful in replacing gas lasers for confocal imaging, LEDs are doing the same and will continue to do the same, I believe, for imaging systems, wide field imaging systems. And it goes back to that confidence in knowing that my students can go to the lab, press a button, turn the system on, and have an immediately stable light source. I don't have to worry about whether they waited for 15 minutes for something to stabilize. I don't have to worry about them trucking back down the corridor and saying to me, the capacitor's blown, or we need to change the bulb, or the image is a bit dimmer today and I don't really know why, and I have to wander down and we have to do an alignment, or we have to change the bulb. That is really essential. And considering that the most expensive commodity that we actually have in the lab is actually people, time. The technology, solid state technology, might be more expensive, but actually in the long run, that isn't our most expensive thing. It's actually most expensive to employ people. So whilst there might be an upfront cost to putting in a solid state laser instead of a gas laser, for example, it's so worth it in terms of not wasting that person's time, not having a day of not being able to do experiments. For example, you have all the setup for an experiment. If you can't achieve that experiment, you waste those materials and you waste that person's time. And if you the number of days we used to lose with gas lasers, for example, where we used to send the argon ion tube back for repair, systems would be down for such a long period of time. The advantage you have with something like an arc lamp, of course, is it's extremely bright. You also have generally a white light source, although there might be peaks within that light source. So you do have that ability to have light across the spectrum, but I think with the developments in LEDs now, there's no reason to necessarily go back to needing a white light source, unless your application particularly demands it.
And we've seen developments in LEDs, and Heitz's law is well known, and the Moore's law of LEDs.
Absolutely.
Power is going to continue to increase over time. What do you see for the future of LEDs in your research?
Well, it would be great actually to not have arc lamps in the future. Personally, they are a consumable. They do go down. We've had to change bulbs on many occasions over the years, and we have lost various capacitor boxes over the years that have had to go off for repair. What would be marvellous from our point of view would be to actually have an LED solution across a spectrum, so that we did have that instant on and also the instant switching as well. I think the narrow bandwidth of the LEDs is a particular bonus, that you can actually switch them on and potentially not worry too much about needing specific filters, because the light comes through, rather like a laser line, comes through with a very narrow bandwidth. So you can have a high intensity light source whilst also having a very narrow bandwidth, which means you don't have to change filters, which can be a great advantage. Every time you have to change a filter, so if you have a white light source and you select a particular wavelength of light for excitation using a filter, there's an overhead. It can be a few milliseconds. There are some very rapid filter changes available. There are some very rapid diffraction grating or prism-based systems as well that will very rapidly change light for you, but there is an overhead nevertheless. And I think the microsecond switching of LEDs is potentially a huge advantage. And also the filters don't last forever. They do decline with age. They have special coatings, and you have to be very careful with them. And they are another consumable. So LEDs have the capacity to be an instant on light source that potentially could span the whole spectrum. Might mean you don't need to actually employ excitation filters. And hopefully that will actually greatly simplify a system.
And are you seeing the switching speed as an advantage in Fura 2 microscopy, fluorescence microscopy imaging?
Well, I think for some applications, for some of the cell types we study, speed is an issue.
Yeah.
So particularly in the excitable cell systems that we study, we need, tens of millisecond resolution. And with some filter changes, simply can't do that. So if you want, for example, to use Fura 2 because you want ratiometric capacity, and you've got to excite alternately at 340 and 380 nanometers,
Mm-hmm.
some filter changes will do that quite quickly,
Yeah.
like within 20 milliseconds.
Mm-hmm.
But if you can just go like that with microsecond switching of LEDs, then
Mhm.
that's absolutely perfect. Because you then have the ultimate, speed readout you can. Whether the camera you have can keep up with that is another question.
Mhm.
But there are some fast cameras out there. Whether there are enough photons of light, given the amount of fury you might have inside the cells to excite, that's another question.
Mhm.
But if your illumination system at least provides that speed, then you can start from that basis and you can empirically work forward and say, okay, we've got a system where the illumination can chop at 150 millisecond
Yeah.
rates between 340 and 380. What will our imaging system, what will our cells allow us to read out from that?
Mhm.
Do we have enough Fura in the cells if we use 150 millisecond switching to actually get a sensible signal out of a cell? Can the camera track that as well?
Mhm.
But if you don't have that light source at the beginning, then of course you can't even entertain that kind of question.
Yeah.
So not all applications would need that speed, but if you have it, why not? If you have that capacity, why not have that? Because then when you do encounter a particular biological question where you need that speed, then it's
Yeah.
there for you,
Yeah.
even if it might not be your major kind of a modality.
Mhm. Mhm. Well, it's been fascinating sitting here,
My
and
pleasure.
I think we've gone all the way through the history of recent history of calcium imaging, and it's been fascinating talking to you,
My
Martin.
pleasure.
Thank you very much.
Cheers very much. Thank you.
Thank you.